So it's a real pleasure today to have Brodie Lawson give us a presentation. So before Brodie gets started, let me give him a bit of an introduction. So Brodie is currently a postdoc in the Centre for Data Science, and it's going to be a particular theme of 2021 in the Under the Hood seminar series, because we're going to hear from all of our postdocs this year. So it's going to be a really exciting year. So Brodie obtained his PhD in applied mathematics, and he worked on creating computer simulations of self-perforation and migration. And just before coming to CDS, Brodie was a postdoc in ACEMS, which is the Australian Centre of Excellence for Mathematical and Statistical Frontiers. So Brodie worked on a number of things during this postdoc, but one particular project that he spent quite a lot of time on was bringing together ideas from computational mathematics, Bayesian statistics, computer graphics, and manifold geometry in order to better understand the really challenging research problem of signaling dysfunction in the heart. So actually one of the outputs from this work was a paper that was published in Science Advances, which is actually a very high-ranking journal. And also throughout this work, Brodie's managed to build a very wide network of international collaborators. And so this includes academics from Oxford University in the UK, and he also has other collaborators in Brazil and the Czech Republic. So now I'm going to throw it over to Brodie, and Brodie's going to talk about population data getting to the heart of variability. So as you're watching along, please feel free to put any questions you've got into the Q&A chat window, and then we can address them after the seminar. So I'll hand it over to Brodie now, and thanks very much.
Yeah, thanks a lot, Chris. And I assume people can see the slides now, so I'll jump right in. And I haven't done an outline slide because we kind of need to go on a bit of a journey to get to where we want to get to. But we'll start out nice and simple, just talking about what I mean by population data. And so I've got here, a traditional type of experiment, although you can often think of other data as coming from what is essentially experiments. And the process that we have is, okay, we go and do some experimental process. We take the observations of what we're trying to measure. And, if we're doing a good job, we probably take multiple replicates. So we get multiple pieces of data, and you can see there, they're all varying a bit in terms of what we actually get the individual values to be. So here we're measuring nanoamps for some current. And if we were to think about this, we say, okay, well, we've got some imperfection here. Maybe it's our measurements aren't perfect. Maybe the actual conditions are changing between the experiments because we couldn't control everything properly. Maybe something a bit more fundamental is going on. And this is still in the traditional setting. So this is not really the population data setting yet. And in this setting, we can set that up with this kind of pretty standard statistical model here. We get observations, and they come from some model which converts the parameters of the experiment into the observations, but there's also some error present. And we could go a step further and say, well, there's also individual effects. So maybe you could have things like a hierarchy where you have different groups of experiments that you think behave differently. You might think that individual experiments behave differently, and you want to encode them in these individual effects. So there's a whole sort of school of statistics related to that. Not really my school. And we're going to see that in this case, I don't think we can really use that traditional approach. Okay, but so that's the basic idea. We say, okay, if we have a traditional experiment, we collect some observations which are being filtered through some function, and we might have some, arbitrary error in here, or we might have some individual effects representing what's going on with our individual measurements that we've taken. And what I think is different is when we know that we have a variable population and we strictly care about that variability. So we'll see what I mean by strictly care, but I've just done this little demonstration here to highlight that now this is kind of personalized data. So we've taken lots of observations. It's not all the same process, and we might be doing the same measurement process, but it's effectively not the same process in the sense that our experimental conditions are changing, and we know they're changing. We're not trying to control for it necessarily. And so the big example here is, yeah, well, if you collect measurements from a population, you know, everybody's different. And so you think about medicine, like the examples we're going to use later. In medicine, you know, everyone will have different genetics, which means their body just behaves differently. And so there, the variability is intrinsic to sort of our results. And so now when we get this variable data over here, we don't really want to, you know, encode it just as, oh, it's an error term. So we return to this functional form, or this mathematical expression I've got here, but we say it's not really what we want. What we want is every individual to have their own truth. And so what we're really looking at is all of our observations have come from a different set of parameter values, but we haven't measured those parameter values. So it's not a kind of fixed effect story where it's, oh yeah, we knew all the ages and now we've got the relationship between some medical observation and age. These are unobserved parameters that, you know, could be subtle genetic effects or things like that, that we really need to actually tease out. And it turns into quite a different problem. And part of the little tangent that will go on the way to getting to where we want to get to, we'll be looking at, you know, what makes this problem different and non-traditional in a sense. Okay, but first, why do we want to do this? You know, random effects modeling or fixed effects modeling or mixed effects modeling, they, you know, have a big history of successes. It's very, very common in statistical modeling. But my argument here is that it's not always good enough to do that. And I don't want to poo-poo the method too much because, as I said, it's not really my school of statistics where I've come from. And some of the things that are covered here, maybe you can, you know, adapt that technique to deal with it. But the sort of difference that I think, the key difference here is what we're actually doing when we talk about what is sort of like a random effect. And if you think about the random effect, it's just plussed onto your observations. And so in this example here that I've got, we're going to record a bunch of heights for some balls bouncing. And we've got multiple bounces, I guess. And so the parameters, I've called them, are the ball roundness and the ball, you know, in quotes, bounciness. But we haven't measured them. We don't directly know what they are. We can try and figure them out, but we haven't measured them. And so the problem is if I represent everything in terms of, you know, what was its random, like the individual effect of this ball on height, I have no idea about roundness and bounciness. And I might want to know that because as soon as I change my experimental conditions a little bit, if I don't know the parameters of interest, I can't predict what's going to happen anymore. So we really want to know what are, you know, the bounciness and the, I've already forgotten the other one, the bounciness and the roundness. And so that's quite a contrived example, but we can move on to, you know, a bit more of an illustrative one. Sorry, I'm just checking my timer. A bit more of an illustrative one, which is in medicine. And so I've got this big chunk of text here, but I've highlighted the bits that I found quite, you know, interesting in particular. And so the ARD, which is just kind of a measure of, you know, effect of treatments. And you've got there the ARD for antidepressants versus placebo was only 17%, which is not, you know, a major effect. But that's not saying, oh, antidepressants don't work, because that effect is being averaged over lots and lots of people. And so one of the big challenges, particularly in medicine, although I'm always interested to hear other you know, fields or topic areas where people think these ideas of population calibration could come in handy, but medicine's a really, really big one. And you see it there, because that's not saying, oh, 17%, it's, you know, quite a weak drug. What you're really going to be seeing is a whole bunch of people didn't really improve. A whole other bunch of people improved significantly. You know, like this is a drug that has been tested, gone through the approval process, has some measure of success. And it's not going to be some very weak effect. It's going to be an inconsistent, strong effect in a lot of cases. And the same story down here, out of 17 common pharmacological treatments, only 11 would actually be called a minimal clinically important difference. But again, that doesn't mean, you know, the other 6 common pharmacological treatments are just nonsense. They're useless. They don't have an effect, because these are effect sizes are being measured across, studies of lots and lots of people. And this paper was actually a meta-analysis, so it's, studies of studies. And what it's really saying and highlighting the point is that, yeah, our medicine is just often inconsistent. And a lot of the time, the doctor will say, well, let's try this. Oh, didn't work, let's try this. And sometimes that's because, the condition isn't known. Oh, I'm not sure if it's asthma or another lung problem, for example. But other times it's because we don't know how your individual body is going to respond to this treatment for the asthma that we know you have, is asthma. So this issue of variability is actually really, really critical. And drug efficacy is still, a major, major point. And there's a lot of money here too, in the sense that, there's a lot of money in pharmacology, but there's, rigorous testing procedures, and everyone's chasing, sufficient effect size and minimizing adverse effects and everything like that. And getting good performance in both those metrics relies quite a bit on being able to understand when we get the good effects versus the bad. And so my topic of interest is electrical signaling in the heart, as Chris introduced. And it is a really prime example of this sort of thing, because a lot of the treatments are very inconsistent. It's a very complicated system. So there's a lot of variability between people that has very unpredictable effects. And I've just pulled out two little examples here. So dofetilide is a antiarrhythmic agent. And so if your heart is experiencing certain types of arrhythmia, and I believe this one was treating atrial fibrillation. But if you're experiencing this arrhythmia, you could get an antiarrhythmic drug that essentially tries to change the behavior of your heart, and we'll see some more details of that soon, to stop it from beating, out of synchronicity. But the issue with this one is, and I believe it's not really strongly promoted anymore as a drug, it actually creates arrhythmia. You can see the figure there, 0.3 to 10.5%. So we don't even have a good estimate of, what percent of people are affected by this, because variability just makes everything in experimentation so much harder, which is why it's such a fundamental issue. But yeah, we've got an antiarrhythmic agent that potentially creates arrhythmia. And actually, atrial fibrillation doesn't kill you. It just, increases your risk of other things and lowers your heart's performance. But Tossard de Pointes, that's probably not pronounced quite right, but that one can kill you. So this is actually a very, very serious issue, is understanding, well, when does that happen? And honestly, in this specific case, a lot of experimentation has gone on, and they do have a pretty good understanding of that. But the key issue of variability and how unpredictable it can make things, and inconsistency of drugs, is a really big thing in the heart, as I think I've emphasized enough here. And so this next picture is just showing, and so this is actually from a computer simulated model, but it was calibrated in a population sense, quite crudely. But here, it's actually looking at what happens when a treatment is applied for cells that are, I think it was, and it's not heart failure. Anyway, unhealthy hearts with a specific condition. And what you see is that in our virtual population, after we give this treatment, a lot of the cells just stops behaving as they should. So the blue lines is, okay, the heart is unhealthy here. It is actually sick, but it's doing the normal thing. And we'll talk about what normal is a bit more later. But when we hit with this drug, a lot of them still do the normal thing, but modified as we would like. And then a bunch just kind of lose it. And we really want to know, why does that happen? And under what scenarios does that happen? And it's very difficult to experimentally do this, because as I said, in these experiments, our parameters aren't often measurable. So we really need computer simulation to dig into this. And I have another slide to emphasize that later, but I guess I've already said it. So the first brief detour that you'll have to excuse, but I'll try and go fast, is just what is actually going on in the heart. And I find it quite fascinating, so I do like to talk about it, but yeah, I won't try and spend too much time here. But one of the fundamental things is that all of the, the muscle cells, which are the main cells in the heart that cause it to beat, the way that they work is they wait for a signal, and that signal is electrical. So while your heart is beating, it's actually, electrical waves traveling through it that are telling it to beat. And this little orange curve here is just a curve of the voltage in millivolts over time for a cell that responds to a signal. Okay, so we get a signal, suddenly a bunch of sodium ions are let into the cell, and it races up in this really sharp peak, and then races back down by spitting out potassium ions. And the reason we have all these funny ion behaviors going on in our heart cells is because they need calcium ions to actually signal. So the calcium ion is what tells it, okay, you need to contract. That's what makes it beat. And for that to happen, the calcium channels need to let in calcium ions, and they only open after the voltage has shot up above, certain thresholds. And so you need those other initial ion transports that we just talked through to get that effect. And then it returns to the rest state. And just two things to say about this. First thing, and these are kind of general interest things, they actually use caffeine in a lot of heart cell experiments for the electrophysiology. It causes heart cells to dump all of their calcium. And as I'm explaining here, this ion transport is actually a very complicated process that controls what the heart cells do. So if you ever hear about things like, oh, don't drink too many energy drinks, it might give you heart problems. It's not just like myth, and it might be a rare event, but there's actually a lot of, mathematical and electrophysiological basis for these things. Second thing I'll just say, when you hear people talking about electrolytes, and I know when I heard about, oh yeah, you got to get your sports drink with the electrolytes, and I'm like, this sounds like a bit of a joke. Is that just some fancy word that they've made up? Because a lot of that does happen in advertising, but no, the electrolytes are literally your potassium ions, your calcium ions, your sodium ions. So they, again, control the fundamental behavior of your heartbeat. That's actually very important. And some conditions, like I was reading, preparing these slides, grabbing out some of the medical stuff, alcoholism is one thing that can really, really deplete your potassium. And then there's sort of like a series of heart-related disorders that are essentially caused by alcoholism because it throws out of balance your electrolytes. So that was another thing I was just interested to learn was, okay, no, there is actually something real going on there with, all the sports drink stuff. Anyway, the real point is these heart cells, they're not smart. I mean, they have this very funny, complicated looking curve, and I'll show one of the equations a bit later, but there's a lot of equations underlying some really sophisticated behaviors, but they're not smart in the sense that they're just waiting for a signal. And if they get a signal, they activate and pass that signal to their neighbors. Then they take in the calcium ions and contract, and then they return to the rest state and await for the next signal. And the reason they have to wait to go back to rest is to stop them from being triggered again too quickly. And we don't want them to be triggered again too quickly for the reason we're going to see on the next slide. But so yeah, in the healthy situation, this little node, kind of at the top of the heart-ish, don't quote me on exactly where it is, sends these electrical pulses through the heart tissue. And because it moves through it as a wave, it causes a synchronized beating motion like you would want. Problem is, because the heart cells, they don't know whether a signal has come from that little node or whether you've got something else going on like we'll see in these videos. And so this mindlessness of the heart cells is actually a really fundamental issue. And so if we just watch through this, this was some simulations that we did. And what you're seeing there is, electrical waves moving through the heart. And we also had some kind of like blocked up tissue in this scenario. But that's kind of much of a muchness as far as this talk is concerned. And so all we're doing is just stimulating the heart, sending those pulses through it. But we're doing so quite quickly and with those blockages in there. And what you actually see is that although all the pulses are starting here, we now have actually created a self-exciting structure. So that's what you're seeing now. You're seeing these spiral waves. I've stopped the pulses coming from that other spot, and the heart is still beating. And it's beating however fast that wave rotates, which, by the way, is usually much faster than what that little node wants to beat the heart. So that little spiraling pattern that you see at the end there, that's your heart being triggered again and again and again very, very quickly. It's called tachycardia, which just means a fast heartbeat. But that has lots of bad effects for your heart because it means that it's overstressed, which can then cause further problems and exacerbate it worse. And in the second example here, I show just how it can go even more wrong. This is now just a 2D simulation in tissue that we did. And we artificially create the spiral pattern from the beginning. But you can watch what happens. And so firstly, you see, yes, we just have those dynamics of waiting to recover and then getting excited again. And then we can create a spiral that self-excites. But you see that it wanders around a lot more than in the other example. And you also see other little waves start to appear where you get what they call wave breakup. And so this is actually now tissue that isn't just being paced really, really fast, but still by waves that kind of do it consistently. Now it's being, triggered chaotically. And what that means, if you have that in the main chamber of your heart, is it's no longer doing this, very synchronized beating motion. It's just beating randomly everywhere. In other words, it's non-functional. And so that's fibrillation, they call it. And if you have that in the main chamber, which is the ventricles, you have sort of minutes to live, which is why they have defibrillators placed, around in lots of places. And if you weren't familiar, and I mean, if you've been through some of my talks before, you've probably heard this stuff, but another kind of common interest thing that I was interested to learn is that defibrillation works just by resetting the electrical signaling of the heart. So that's why they, jolt with electrical pads in hospitals and things like that, is because what you're doing is you're saying, no, I don't want to spiral there. Just reset the whole heart. Set it all excited and then all relaxed together. And now the node will be what starts it going again because it's pacing by itself automatically. So that's actually how defibrillation works on a sort of most basic level. Okay. And so this is the slide I was saying, it just kind of re-emphasizes the point, which is, yeah, you've seen now how complicated the dynamics are, and we can't measure the parameters that are controlling those dynamics, our thetas. But we know that those theta values do vary a lot between people. So you can take measurements of individual heart cells or individual hearts and see that they behave very differently. And so this is why we really need computer simulation to investigate the effect of that theta. But if we want to investigate the effect of that theta, we do really want our model to be representing reality. So we do need to take that very variable population data and calibrate to it, which is what the talk is about. And so this is the next thing that's kind of a quick aside, but just for people who are interested in the mathematics behind it, what we're looking at, you actually just simulate the heart cell as a capacitor. So this is just your capacitance and then the rate of change of voltage. So if you have your Q equals CV from high school physics and you take the derivative of it, you get this, but we have lots of currents, which is all of those ion flows in and out of the membrane of the heart cell. And I've shown here just one example current just to make the point of what we're actually dealing with when we talk about our mathematical functions and the complexity of the system. So this is the sodium current, the main one that lets in this big spike. And you have a strength of the current, which is actually our parameter theta. So we have lots of current, so we have lots of theta, different components of theta as a vector, but that's the main parameter, is the strength of these currents. But then we also have these gating variables that tell us what's the chance that these channels are actually open. And the way that it works is it pushes towards the Nernst equilibrium. If that doesn't mean anything to you, yeah, it doesn't matter. But it's kind of just for people's interest who may have a bit more of a physics background. But last thing I'll just say, we have this voltage dependent behavior that's quite sophisticated. These are actually just curve fit from experiments, but you have these messy functions in here that tell you how much your M channel, in this case, likes to be open or closed depending on the voltage. And so that's the complicated control that the heart cell has over which channels are open or closed. But as I've highlighted before, it's still mindless in a different sense. Okay, we won't waste any more time on that, but I will quickly say when we take it to tissue, so here I should have said this one is explaining this dynamic here. And so it's an ODE system because we have the change of voltage over time. That's also then driven by the change of these gating variables over time. But when we take it into tissue to get these spiral wave dynamics, what we have is a reaction diffusion system. And so you just have this extra diffusion term, which represents the spread of excitation to neighboring cells. And then here you have all of those currents that we just talked through on the previous slide that give you the excitation behavior and the refractoriness behavior, which is, you know, the time it takes to recover. So yeah, we have really complicated dynamics, but we have them pretty well mathematically described. So it's a very good setting to then explore variability in the computational setting. And so now finally, we come to the population calibration. And what I've done here is I've come back to the population data slide where we said, well, we want to know everyone's individual truth. I'm going to do a step further and say, I don't care about error for now. That's something that we can look at, but we can also get a lot of gains without even touching. So let's just look at this. We've got yi equals f of theta I. And we know what f is. So you might say, well, you know, can't I just go and solve that or try and do a minimization to get as close as possible in my model? But we don't want to match every individual observation to some individual set of parameters. Because firstly, multiple parameters might give me the same observation. And secondly, we want the spread across the whole population. If we want to go build a virtual population to do experiments on, we want, you know, a representative sample of all the people. We don't just want, you know, the 15 points of data that we have, say. So we don't want to over depend on individual data. We want to get a spread, which is indicated here by saying, yeah, if I've got a distribution measured for y, these are my observations, how do I convert that into a distribution for theta? And so you might say, oh, you want to get a distribution from some data, that sounds like Bayes to me. And unlike most other Bayesian talks that you go to, I'm not going to spend the slide introducing this formula. I'm just going to talk about why it's a bit dodgy here. So this is saying, well, what's the chance of seeing the data given some parameter values? But my data is from lots of different people in the population. I don't have one set of parameter values. I can't tell you the chance of seeing all my data given one set of parameter values because I know that that data came from multiple sets of parameter values. So I'm like, maybe I can't do that. Or maybe I can do something like this. And so now if you are familiar with, you know, Bayesian statistics, you're probably screwing up your face because that just looks all kinds of wrong. But if you're not familiar with Bayesian statistics, maybe you'll believe this a little bit more, is to say, well, if we just weight by how likely we were to see that data, and this was our distribution of y that we're trying to recapture in our population, we're saying, oh, if a theta value gives me y down here, well, that's not very common in the population, so I don't want to accept that very often. But when I do my sampling and I'm up here, so I've got theta values giving me these y values, I go, oh, yeah, actually that looks pretty good. I'll take lots of those. So if you squint, you might say, oh, hang on, maybe Bayesian stuff can work here. And I've gone down a bit of a philosophical rabbit hole about that. I call it philosophical, but it turned out to be mathematical and just wrong, as we'll see. So here I've got a very simplified example, and I've just got, you know, two types of people, big people and small people, and we've got two DNA elements, so you know your CG, AT, and if you have this specific location in your DNA strand is CG, turns out you're going to be small. Otherwise, you're going to be big. And we can look at the population and say, oh, 40% of them are small. We can easily measure that, but we can't easily take people's DNA and check those two genes, or not even genes, just DNA pairs. So what we're saying though mathematically is, okay, I know my probability of observation is 0.4 small, 0.6 not small. And I want to know, so what is my distribution of the parameters based on that? And I'm going to go a step further and say, let's have a uniform prior. So outside of, you know, our observations about who's small and who's big, we have no information about those two genes. So they're all, you know, 1 on 16 chance because there's 16 combinations we can have for the two DNA pairs. And so if you set aside the maths, maybe you can see the answer already. And if you're switched on, maybe you can also see why the Bayesian method doesn't work here. but it took some thinking for me to properly think through. But yeah, if we don't touch any mathematical notation, we say, well, 40% of people are small, so they must have CG, right? And the other 60% of people, well, we have no information about what they have other than it's not CG. So we should just distribute that 60% of, you know, people across those genes evenly. And that will give me my spread of what people have for their genes, sorry, for their DNA, over the whole population, which is great. But if we do it from a Bayesian perspective, what we're now looking at is, okay, I generate a random DNA pair. That's my parameters theta. I stick it in my model, and it says, yep, okay, what's your Y? And then I'm going to go accept that based on population frequency, which was my kind of, you know, in quotes Bayesian thing. Maybe it'll work that I showed before. We just say, you know, is this a common thing in the population? Is it not? Let's accept it or reject it based on that. And so we end up sampling lots of non-CG pairs because CG is only 1 out of 16 combinations. And every time we sample one of them, we say, well, we get a big person when we put it in the model, because that's what, you know, any non-CG pair gives me a big person. Then I say, okay, how frequent are big people in the population? 60%. Oh, that's pretty frequent. So I'm going to take plenty of those samples. And so if you follow that through and then normalize your posterior, this is what you get. So this naive Bayesian approach will give you a population where only 4.3% of people are small instead of 40. And the reason for that is because we haven't taken into account the bias that comes from, oh, well, tons of my theta values tend to give me big people. And it's because the Bayesian perspective looks at it kind of backwards. I call it backwards, but, you know, really the Bayesian thing is just saying, well, tell me the likelihood of the parameters, and I'll filter it through to be a posterior for you. And we're doing something a bit different. But we can actually mathematically attack this. And in this simple example, it's not too bad, because you say, well, this is just a change of variables. My chance of seeing something in the population is just the chance of having some theta value that gives me that outcome in the population. And I've got to add up those probabilities for that set of people that does it. And so if we have a uniform prior, we can go a step further and rearrange this and just say, well, the chance of any given set of parameter values is just, you know, what proportion of the population gives me that outcome, but then I've got to divide it evenly across all of the different, you know, options for theta. So in this case, it's like, oh, I got 60% of people are big, but I've got to go divide that across the 15 pairs of genes that can possibly give you a big person. And so that's just the intuition that I went through before, but expressed mathematically. Unfortunately, taking that to multivariate and continuous variables is a lot less pleasant. But I found this author who, you know, the paper that I'm quoting, the 2017 one, has 10 citations, but I think all 10 of them might have been him. But it's actually a really awesome paper. And so what it does is give us a tool for expressing mathematically what we want that avoids measure theory. Because I think maybe measure theory can do this, but I don't know measure theory. So what we instead say is, well, we have a proportion of population demonstrating the outcome. So it's just an integral with the delta function being, oh, f of theta matched to y, because the delta function is only non-zero when we have a match between these two. So that grabs out that outcome, and then p of y is how likely that outcome is. And then here we need to divide it across all of the different options for theta that could have given us that y, the same intuition we just chalked through. And we do that by saying, well, let's integrate over all the thetas and then grab out the ones that give us that y. So yes, this actually is a mathematical way we can do exactly what we want. But it's two multidimensional integrals, and we don't know how f of theta really behaves. You know, it's some complicated heart model or whatever it is. So we can't just naturally jump to anything from this. So we have this nice framework, but does it do much for us? And so I say, well, approach one, we give up on that. We go back to the Bayesian in quotes way that we just showed doesn't work. Or step two, we use a simplified f of theta to attack this. Okay, so if we go back to Bayesian, I've just recreated the figure there, but what we're going to do is we're going to use a, you know, nice Bayesian sampling method, because it will at least give us this intuition of, well, if lots of things in the population have that value, then I definitely want more of those. Maybe we're going to be biased by, you know, how many thetas give us that y, but at least we're getting y's that are the good y's. And so I was using SMC. This is the work that Chris and I and a few other people were on, the one that was in Science Advances. So we took it and we said, well, let's do SMC. That's a good sampling method. I've laid out how the sampling method kind of thinks here. And I've also then done, the mathematical version of it. But I think, I won't talk through that in the main talk, just in the interest of time. But it's a sampling method that I'll just give the really brief overview here. Yeah, so you get a bunch of particles, and then we say, okay, well, my particles just came from my prior. They're probably junk, but I'm going to check how junk are they. And based on that, I'm going to say, well, how much, of the real problem am I actually solving here? Because I've solved the prior. I sampled from the prior. Am I sampling from the actual distribution I wanted to get this thing? No, but I'm getting a little bit of the way there. And so then I'll introduce a bit of that and then copy the particles. So now I'll lose a couple of particles, but they'll be the better particles. Will be biased towards that. And how strongly I bias it is based on, how safe it is to introduce this complexity. And then I'm going to give each particle a little bit of a wobble so that they find unique spots for themselves again, because otherwise they're all bunched on top of each other because I just copied them on top of each other. Okay, and so we just keep doing that until we are able to safely include all of the real sampling problems complexity, and that gives us a sample from our distribution. As we said though, this is the Bayesian in quotes method for our purposes, so it's not technically the distribution that we want. And so we did a post-processing step to repair it. What we do is we just sample, a big number of particles using SMC. And then we say, well, all of those particles, I know what their outputs are. Like I know what, I've got theta values for them, but I also know what the associated Y values are. So I can check, oh, if I take some subset of that population, I can maybe get an even better fit to the data in a distributional sense. And so that's what we did. We defined some measure of goodness, rho, and then we said, okay, let's just try randomly including or removing particles from our full set to create this subpopulation. And I'm going to accept or reject them with a Metropolis-type algorithm here. This is also simulated annealing, but with a fixed temperature for people who know that buzzword instead. But this is just, post-processing, we pick an optimal subpopulation. And it's nice because, we have all the Y's and the thetas. So it doesn't take us a lot of time to do this. The only time is if rho is, costly to calculate because you have to try and measure differences between distributions. And we'll see it in action. So, if that slide was a bit too fast, you can at least see here the idea in effect. So the red dots are a poor calibration method, which is kind of what people were doing in literature before. The blue is what you get from SMC without fixing anything, and the white is the data. So you can see out of like our whole square, we are honing in on the data, but we've also got a lot of junk. You can see in a lot of spots. One other thing to point out, our model was never actually able to recreate the data here. So no matter what you threw at it, it only ever sat on this one little line. So that is just kind of highlighting that your model and the parameters that you say are variable should be able to actually capture the variability in your population, something that you need to think about. But yeah, we've got, better than the red, and we're getting towards what we would want. And then we come along and post-process. And so now the blue in the previous slide is now turned into the red in the background. And now we have the light blue versus the white. And you can see, other than this one and the bivariate plots that involve it, so like things like this, we actually have a really good fit now. So this post-processing does do what we want. Thing is, SMC, you're taking a lot of samples. So if your model takes a long time to run, might not be fast enough. And so that's when we come back to approach 2. And so in approach 2, what we're going to say, well, got rid of the prior here. But if we look at this and we actually pretend we know, when is this delta term actually non-zero? So like when do we have y equals f of theta? We could actually just sample from P of y and then sample from the set of theta values uniformly that give us that y value. And that would do the population calibration absolutely correctly. That would do, this Paul Bagenstoss's guy's approach to how we should express it. And if we have a linear model, we actually can do that. So we say, all right, we've got some linear relationship between y and theta now. And so if I have some given y, I can rearrange to get theta by itself just using the pseudo inverse of a, because a is rectangular, because you don't have any guarantee that you have the same number of measurable outputs as inputs. But with this theta value, I can then say, and technically, if A is, if it has a null space, then I could move anywhere in that null space and I'd still be on the correct value. So I have to add in, an arbitrary vector from this null space to get the whole manifold. But so that's great. Like that, I actually have a solution. And I can sample from that manifold in the bounded setting without any kind of trouble. It's covered in his paper and it's not hard to code up or anything. But is our model linear? Probably not. Let's be honest. I mean, I just talked about how complicated heart cells are. And now I'm going to come tell you, oh yeah, but the observables that we measure, yeah, they're linear related to the inputs, which are those strengths of individual currents that behave with all those funny exponentials curve fit from experimental data. Totally, that's linear. And yeah, you probably start, again, screwing up your face. But what we can do is at least build the best fit linear model. And so what you're looking at here, the S's just indicate that it's standardized. And then we have just the least squares solution, but with Tikhonov regularization in here, or ridge regression, whatever you prefer to call it. So we can get the best fit A, in a sense, like, the best linear model that tells us how a set of theta values relates to a set of Y values. How do we get those sets? Well, let's just, get some particles and sample them from the prior, which is just uniform across our parameter space of interest. So we can get some approximate A for our model. Still might not be that good. And it might especially, if we're looking at particles across the whole space, no guarantee that how the model behaves in a, mean field across the whole space sense matches how it behaves in the region we want to calibrate to. So how do we address that? Well, the idea was we can just build better and better linear models as we go. And the way that we do that is when we have the crappy initial linear models, if they're going to do bad for us, we just won't trust them very much. So if we don't trust our linear model, only a little bit, so whatever it proposes, we just weight that proposal by S and keep the old location with the rest of, 1 minus S. So if we got a bad linear model, well, we'll just take a little bit of S. And hopefully that will just push us in the right global direction until we can start building better linear models. And so how do we select S? Well, we want to know, yeah, am I getting better population calibration? So am I getting a better distributional fit? How do we measure distributional fit? Same way we always do. Kohl-Black-Leibler divergence. You could do other more sophisticated ones like Jensen-Shannon. And in the Science Advances paper where we did the SMC approach, we were using something based on Jensen-Shannon. But in this case, we did things very bare bones. So it is using sort of moment matching and assumptions of normality, which gives us an analytic form for the Kohl-Black-Leibler. So that's the one we were using in this case. And so here it is on a toy problem. And this is quite a nasty toy problem in the sense that it's definitely not linear. I mean, you can see that it's not a linear function there, the model. And you can also see you've only got 2 inputs, but 3 outputs. So you don't actually have, enough degrees of freedom to fully explore your space of data. That said, we're going to use synthetic data that did come from the model. So technically it should be able to recreate it if we can calibrate correctly. But this is supposed to be a pre-testing example. The weird thing is it turned out not to be a very testing example. So here we see the initial start. And so you just have a 3D view that is 2D, sorry. And then you have 2D slices, the 3 different combinations of parameters. But what we actually see is, okay, the linear model doesn't look very good. But if it's not good, maybe we just won't trust it too much. We just want to suggest better things. So our red things that we start with aren't good, but after 1 iteration, that's what we get. And now the linear model that we're going to build for the next iteration is sitting right along the area of interest and capturing kind of the overall slope in that spot. And even though we're using a linear model to propose these things, because we're still filtering through the model itself, the real model, every time we take a theta and stick it in and get a y, that's using the real model, which is not linear. So we can actually still capture the nonlinear behavior. And we see that, it does a really good job. The KL is tiny. Great. Here's an example on real data, not as impressive, but you've got to remember that we're comparing this to the SMC. We haven't done any refinement here, so this is the unrefined SMC. And so you can see while the SMC on the previous slide, a while back, we saw that before we refined it, it kind of still blobbed out everywhere. This one maybe hones in a little bit too much, but this actually very rapidly, like this is where it starts at, very rapidly actually finds the region of calibration interest and does kind of capture the overall spread of it reasonably well. And what we see, just checking the time, what we see is that it performs much better in terms of efficiency and performance on this example. Okay, you're probably getting freaked out by this red spike. All that's happening there is I'm not plotting the best over the whole course of the algorithm, which is normally what you would use. I'm just showing the actual performance as it goes. And so the linear one, which is the red line, is spiking up because once it gets stuck in a very good solution, like kind of the best it can do, eventually that line search step breaks down because it can't improve any further. And so that it will end up taking a very silly step and then instantly repair it again on the next thing. So that's what you see here. But yeah, so you can see if I do SMC with a small number of particles, I don't actually get amazing performance, and it's inconsistent because we're targeting the wrong thing. The linear method is targeting the right thing, but crudely, because it's using a linear approximation. So it actually outperforms SMC in terms of distribution or fit, and then also in terms of speed. So great. And now I just wanted to show, to kind of finish us off, what are the actual benefits of doing this population calibration? So I kind of motivated it earlier, but let's just show some of our actual results. So these ones are from the Science Advances example, but the linear calibration is essentially a methodological possible improvement and definitely a much more kind of quick and dirty way to get it done. But in terms of the benefits, we got some really nice results in that original paper. And so what you see here, the blue is healthy cells and the red are chronically atrial fibrillation cells. So they've adapted to an unhealthy heart that's being paced too quickly, essentially. And so what we actually saw was if we calibrate our model to these two different data sets separately and then create virtual populations, which are these red and the blue, we actually capture the key features of the two populations. So the blue, the healthy one, is supposed to spike up, rapidly repolarize, and then slowly return to rest. The atrial fibrillation one doesn't really rapidly repolarize, it does it slower, but it also never actually plateaus, or not to any significant degree, and ends up repolarizing back to rest quicker. And that's why atrial fibrillation is really dangerous for your heart, because if you think about those spirals, if it repolarizes quicker, it's ready to excite again quicker. So the spiral has more chance of running into its tail and finding the tail ready to be excited again. So we're capturing those key effects just by calibrating to the data. Even more surprising, if we go then look at the individual experiments where people go and measure what are the individual differences in a single current between people that are healthy and people with chronic atrial fibrillation, what we actually found was that the computer calibrated population did a very, very good job of picking up most of those without having any of that information. So like I said, people can't measure all the thetas, these current strengths themselves. They can maybe measure one at a time with really invasive experiments, but we were actually able to predict most of those changes associated with the unhealthy hearts just by calibrating our computer model in the population sense. But you did have to do the good population, properly capturing the distributions to achieve that. And the last thing here is just reiterating the point. Oh, the lines are a bit faint, sorry. But so here I was just testing a drug treatment on our calibrated population. And so we found that we had a better predictive effect of what the drug would do. So you can see it's prolonging how long it takes to get back to rest, which is good in terms of not getting spirals that trigger your heart when you don't want it to trigger. But we also see a few members of the population that do some very bad things here. And we have to then wonder, is that a real effect? What set of parameter values cause that to happen? This is something we can investigate. So those are kind of the main reasons that we want to look at population calibration. And so I'm just going to finish as usual with the future directions. Really need to publish the iterated linear calibrator, which was like the second method that we talked through here. I've also got some ideas for how do we do better at matching distributions in higher dimensions. We could also look at post-processing, so that helped a lot with the SMC. No reason we can't do that in the linear calibrator setting as well. as I said, the post-processing step is usually cheap because you've already run the model and got the particles ready to go. So could be a way to repair the over-convergence of the linear one if we, like, for example, inject extra variance, but then smartly take only the extra variance particles that we want. And of course, I'm always just interested in where this can otherwise be applied. It's had a lot of success in the heart area. There's a famous paper where they showed that, computer calibrated populations outperformed animal studies. So it kind of was like, well, let's just use computers and not harm animals while we're doing these heart tests, because animal hearts aren't really human hearts anyway. But I'd really like to know other examples where people think, they might be able to apply this. And just highlighting that last point. So that's roughly about the time I was aiming for. And I've just got here, a list of thank yous for the people that have been involved in essentially these two projects. And there's other people in other projects that might not be acknowledged here, but thank you to everyone. And thank you for coming along and listening.
Thanks very much, Brodie. That was really interesting. So we do have a couple of questions that have come through on the chat. So let me just open those up. So for the first question, and I think it goes back to one of the earlier slides, can your theta i's be from different parameter spaces? So maybe just answer that question first.
Can your theta
so this is back on one of the earlier slides.
Okay, so can the theta i's be from different parameter spaces? So I'm wondering what is precisely meant by different parameter spaces here. Because when I talk about theta i's, I'm talking about like in a vectorized sense, essentially. So I have some big multi-dimensional parameter space. If you mean different populations, you can calibrate to separate populations separately, which is what we did in exploring, the difference between healthy hearts and unhealthy hearts. And so you could always change the initial parameter space you use based on prior beliefs if you think you already know some things about unhealthy versus healthy, for example. But we found we could just take a very, very permissive parameter space that allowed lots of parameters and still get good calibration to the different data sets by just narrowing in on what the data was actually telling us. I'm not sure if that's what you meant by the question, but
yeah, no, that sounds quite reasonable to me. So then a follow-up question from that one is, so I think you're dealing with the deterministic model in your talk. Can it be extended to cases where the model is stochastic? So for example, a stochastic differential equation.
Yeah. So I did just completely cross out the error term. And so we haven't even addressed the case where the errors are nicely behaved here. But at the same time, you kind of capture the error by calibrating across a whole population, which gives you variability naturally. I do still think, though, that there could be a lot of benefit to bringing back in some error. We haven't looked at an SDEs, and I don't want to speak too much about especially the intrinsic stochasticity. I suspect, though, that you probably can do still the work in this area, but you probably lose the kind of neat mathematical form that I showed with the multiple integrals. That said, if you have just normally distributed errors that you add on the top at the end, I suspect you can actually build them right into that normal mathematical framework and proceed quite well.
Okay. So just another question there. So this is about the refinement step. Do you think it describes the tails as well as before? And how do you avoid overfitting your population data?
So when it comes to fitting tails, I guess it really comes down to how well are you estimating the distribution. So one thing I didn't really address here beyond just future directions at the end is you need some way of converting your data into a distribution in the first place. And so when you measure discrepancy between distributions, if you calculate the discrepancy accurately and well, you should still be also capturing tail effects, but that's assuming both that you're estimating the discrepancy well and you're estimating the original distribution well. And so there is definitely a real point there about, well, if I only have a small amount of data, for example, how do I know you know, what kernel bandwidth should I inject if I'm going to do a kernel density estimate to give me these distributions to calibrate to in the first point. And it really then feels to me kind of like more like a modeling choice that you would need to justify. The method itself is quite agnostic to it, assuming, yeah, you're calculating the discrepancies between distributions well, but you do need to think about, well, how do I estimate the distribution in the first place in a good way? And what I found in the Science Advances work, kernel density estimate actually worked really well compared to some other things we tried, like copulars. But when I tried some kernel density type stuff in the linear one, it wasn't doing very well. And I'm kind of thinking about coming back to try copulars in the linear calibrator setting and some of its problems as, you know, an optional future thing. But I think that's really what comes down to whether you overfit or not to the population data.
Okay. So I did have one question actually. So you mentioned about possibly applying the refinement step to the linear approximation approach. So
yeah.
Is the point of this trying to correct for the linear approximation? Is that the point of doing the refinement? Because I thought the way that that one solves it is you kind of solve it exactly, so you wouldn't need to refine it. But maybe you do need to refine it because the linear approximation hasn't worked as well as you wanted it to work or something.
Yeah, that's right. The intuition behind it is, well, if we have a data that's quite concentrated somewhere, maybe if we zoom in on that spot, it looks approximately linear, you know, the classic linearization of a nonlinear function type idea. But especially if your data is quite spread, you don't think that's necessarily going to hold as well. And because you still filter it through the model, the real model, often you get away with it anyway, as I kind of showed in the example. Like that wasn't a hand-picked example. That was just, I tried that toy example and I'm like, wow, it actually does really well. But yeah, there is definitely scenarios, and you see it in the real data example where we have a tougher problem. Probably that data is actually more close to linear, and it looks, you know, at least in the bivariate scatter plots, better behaved, but the linearity isn't quite there, and you can see that the method does struggle a bit. And really what the benefit is you can use it to just very quickly hone in on the area of interest, and then maybe you don't get enough variance, or maybe it still looks a bit dodgy, but if you're in the right area and you have an idea of kind of like, well, my particles seem to think these parameters are important or these ones aren't, so I can just inject variance around that, sample a new set of particles once, and then take some optimal subpopulation from that. And so I haven't explored that in terms of efficiency, but I think it would be quite a, you know, a reasonable thing to do. And I just saw in the chat, there's also a question, what are the boundary conditions of the membrane potential map? Does it wrap around? I didn't go through boundary conditions because I'm a pretend mathematician, but the story is, in my scenarios, it was just no flux on the boundary. So you're just assuming that it's kind of like a wall outside of that. That said, periodic boundary conditions are quite common. And if you look at the heart chambers, they are kind of like a, you know, not spherical, but a deformed ball-like surface. So yeah, periodic boundary conditions are quite an often done thing as well. But the simulation I showed in 2D, that was just no flux on the boundaries, David.
Excellent. Okay, so I think we've run out of time now. So thanks a lot again, Brodie, for the really great presentation. So we can all kind of join together and giving you a virtual applause there. So thanks very much for that.
Yeah, thanks.
And thanks very much, everyone, for coming along today. And we hope to see you next time.
Yeah, thanks a lot.